Hello, I hope you are watching this or safe in confinement. So today I'm going to talk about the raging COVID-19 pandemic and the proposed measurements to tackle it. The main star here is of course severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2, originated in Wuhan back in December that spread wildfire to the whole world with nearly 2 billion confirmed cases. SARS-CoV-2 has been dominating the world, and so much so World Health Organization has to declare this a world pandemic as of 11 March. Now we all know that SARS-CoV-2 is a novel pathogen to humans, meaning we have never encountered the exact same virus in history. The symptoms of COVID includes fever and primarily respiratory distress, and the extent of the severity includes of course hospitalization and requiring mechanical ventilation to support breathing. Some patients are stricken severely to being on their deathbed, while there are some of us who manage to be just fine and manifest just mild symptoms or even be on their way to recovery. We may argue that lifestyle factors, diet, or underlying medical conditions could play a role in contracting the disease. Going back to a central idea, SARS-CoV-2 is a novel pathogen to humans, and so we have never established any antibodies or any prior immunity beforehand. And it's the reason why we've gotten so sick because there's no cure thus far. So the question is, why and how some of us are hit hard by COVID, and why some of us are okay? Scientists postulate that our genetic makeup could have some answers. For that, measurements are taken to determine the truth of this statement. Scientists are proposing that research based on using the genome-wide association studies and the popular approach used in genetic research to link specific genetic variations with the disease in a population. So here is how we do it, and this is the overview of the proposed methodology. We first start with phenotypic identification separated into two phases. The initial phase aims to recruit participants and the second phase to define cases and control. Barriers are seen in both phases, where in the initial phase there is a lack of serological testing, a lacking of community's awareness towards novel infections, and just to find a number of human subjects. If the initial phase fails, this will hinder the progression into the second phase or just stopping the entire research at its starting point. However, if both succeeds, the whole research continues. WHO even proposed the ordinal scale for clinical improvement to facilitate the grouping of patients according to their designated phenotypes. Higher scores implies higher mortality status. To determine the relationship between phenotype and genotype, we should take in mind that phenotype should be collected on the basis of its clarity, quantity, and reproducibility, whereas for genotype, we should have some knowledge on the virus life cycle and knowing some literature on genetics and novel infections thus far, and as well as observing manifestations in patients. Onto subject recruitment, participants for cases should fulfill the criteria of being either hospitalized or symptomatic, or tested positive for COVID or asymptomatic. And for the control group, just a random volunteer will do. The targeted number of subjects is set to be 1,000 or so because a larger sample size have a better coverage in general. Ethical approval are then sought from participants who join on a voluntary basis because this study concerns on human rights. Samples are then collected from nasopharyngeal swabs or any biological fluids where available. The samples are then sent for DNA extraction, and it should be stored under cold temperatures if not in use because DNA are prone to denaturation. Genotyping is then conducted to determine the single nucleotide polymorphisms or SNPs using Illumina's latest technology. SNPs are genetic variants that are different in every individual, and this might be the reason why everyone has different susceptibility towards COVID. In comparison to whole genome sequencing, array-based genotyping is chosen for many reasons, but mainly it is suited for analyzing common and low-frequency variants, which is what this study is aiming to achieve. The first step for genotyping is shearing, where double-stranded DNA undergoes sonication to single-stranded DNA. DNA sequencing is then performed on microarray plates. Beads are attached to the wells, and probes with fluorescent tags are attached to the beads. If a DNA is able to bind to these probes, it implies a complementary hybridization. So using this concept, single-stranded DNA are added in, binds to the probe in a complementary fashion, and excess materials are washed out. Signals are produced once the lasers run through it, which we then analyze and interpret the results. Imputation then follows, which basically aims to cover the missing genotypes from the analysis earlier. This is performed by Michigan Imputation Server, a software that refers to data from reference panels such as 1000 Genome Project. Quality control is followed. For the first will be sample quality, where we focus on the marker quality. The genotyping call rate or genotyping efficiency will be evaluated, and setting a threshold is recommended. However, this relies on certain factors such as the sample quality of DNA. The software Plink will help in confirming this efficiency. The next will be batch effect analysis, referring to undesired variations arising from technical errors. In other words, these variations are biases that ideally should not be happening because samples are plated randomly and we're using high throughput technologies. However, if such outcomes is inevitable, we can minimize this using the software Plink that helps in studying the associations in the whole genome. Statistical analysis will be the last step where we analyze the genetic correlations between the phenotypes. This is performed by TreeWAS, a database designed based on the Bayesian approach to estimate genetic risk. Here's an illustration of the idea following the Markov process, where the genetic variations are arranged in a hierarchical structure for analysis. The white nodes here represents no genetic associations with the clinical phenotype, while the colored ones represent otherwise. The TreeWAS database is preferred here as it offers a distinct separation between the desired and undesired properties, and it uses better sensitivity and specificity over conventional methods. But the users may need a prior programming knowledge before using this. Genetic variants are then identified, followed by meta-analysis. We look into multiple studies to statistically assess a combination of data based on the identified genetic variants. So this is performed to determine the validity and significance of the study. And like the steps earlier, this is aided by software like Stata or Metal. As far as the research is concerned, some findings have been reported. A mutation has been observed in symptomatic patients, the genomic position 11083 and non-structured protein 6, and scientists named this the 11083GT mutation. This is significant in the sense that it downregulates autophagy in host cells, allowing viral infected cells to live, and is also responsible partially for cytokine storm as evident in the increased TNF-alpha levels. But fortunately, the mutation might imply a biomarker and direct a development in drug therapy. As with all approaches, GWAS comes with its pros and cons, but just to name a few, GWAS helps in the discovery of new variant trait associations and novel biological mechanisms, but it is much tedious to perform and may not always obtain the expected outcome. Let's head back to the postulations earlier. Could our genes really do hold some answers to our question in mind? Well, yes, it is possible. Research are still ongoing, but the future still holds out hopes that when the pandemic ends one day, we'll embrace each other and say we made it. Till then, stay home, stay safe, and thank you for watching.